Hi, welcome back to the channel. Today I'm observing plant cells under the Olympus CX23 microscope. I'm going to be looking at 3 different microscope slides, which is the corn stem, onion epidermis, and the pine root. And I'm going to be looking for different parts of the plant cells like epidermis, xylem, phloem, and vascular bundle. Without further ado, let's observe it under the microscope. So there's a high chance that what I'm about to say is wrong because I'm not a biology guru, as you may able to tell. I'm giving this voice over thinking a try, so hopefully I sound real pro. The outer edge is probably the epidermis, and if we take a closer look, we can see the xylem and the phloem. The large hollow cells in the middle that have different colors are the phloem cells. The reason why they are in different colors are because they are stained with different types of stains. The purple smaller boxes are the xylem cells, and they are usually much smaller than the phloem cells. With 400 times magnification, you can see a lot more detail and structures of what's going on inside the phloem cells. Now I'm switching to another specimen. This is the corn stem. Now to one of the most popular specimens to observe under the microscope of all time is the onion epidermis. And it's in the name that epidermis, so clearly this is the epidermis of the onion. But you can also see the nucleus. The nucleus is very nicely stained with the prepared slide. The black dots in the middle of the cell. I'm now using some digital zoom to let you see more clearly. And I think that's it. Thanks for watching. Bye.